Hello friends, myself Professor Namdas, Department of Botany, HM College, Karad. Today we are talking about the nucleic acid, Watson and Crick model of the DNA structure. In this topic, we have planned to study what is the work of Watson and Crick about the DNA model, then what are the characteristics of the DNA model proposed by Watson and Crick, and forms of the DNA. What are the different forms other than Watson, the different scientists have proposed? When we look into the structure of the DNA model, Watson and Crick proposed the DNA model. The model is explained in one line, DNA double helix model. But before that, in 1952, a pair of scientists, Maurice Wilkins and Rosalind Franklin, they have taken the picture of nucleic acid by X-ray crystallography. Means, what is the nucleic acid structure is observed by scientist Frederick Mitchell. That substance is exposed to the X-ray and the picture they have collected on X-ray photographic film. This film is studied by Watson and Crick. Means, Watson and Crick observed the picture taken by Maurice Wilkins and Rosalind Franklin, and then afterwards they have explained that line of the DNA model. DNA is a double helix. See here, this Crick, Watson, Wilkins and Franklin, all these have the contribution about the structure of the DNA model. When Watson and Crick explained DNA structure, at that time, that DNA structure they have described by these 11 different points, where they have said DNA is a molecule of double helix. There are different strands. There is a base pairing. There are transverse bars in the DNA model. They have pyrimidine and pyrimidine ratio which is fixed. There is a pairing between purine and pyrimidine. There is a polarity in the strand. There is a phosphodiester linkage on the DNA strand. The strands are antiparallel. Strands are complementary. And some major and minor grooves are here observed on the DNA strand. And there is specific dimension in the DNA strand. It means that some characteristics, some properties are there in the DNA strand, and they are explained by the Watson and Crick. One by one, when we are talking on that properties or characteristics, we come to the first one. According to Watson and Crick, DNA molecule is a double helix. According to Watson and Crick, means Watson and Crick explain that DNA molecule is a double helix. What it means? According to him, according to them, DNA molecule consists of two strands. These two strands are coiled with each other at considering a central imaginary axis. Means there are two strands. These two strands are coiled. They are twisted with each other and it has a central imaginary axis. Then, second characteristics according to Watson and Crick, the structure of the strand. What is the structure of each strand in the DNA? Each strand here has a backbone of sugar and phosphate. Means on every strand there is a sugar and phosphate, and these are the backbone of each strand. The sugar molecules are joined with each other with phosphate bond, and so therefore they result in pairing of purines and pyrimidines. So there is a pairing between one purine to the another pyrimidine. Means adenine to thymine, cytosine to the guanine. And so therefore A is equal to T and G is equal to C. Always there is a pairing with different hydrogen bonds. When A pairs with T, there is a double bond. When guanine pairs with cytosine, there is a triple bond. See here. When we look into this picture, in the base pairing, we find here is A as a purine, here is a T as a pyrimidine. So purine pairs with pyrimidine with this hydrogen bond. If there is a pairing between A to T, there is a double hydrogen bond. Now see into this. When we look into guanine as a purine, cytosine as a pyrimidine, and there is a base pairing, at that time there are 3 bonds. Means guanine to cytosine there is triple bond. Now, in the DNA structure, there are transverse bars. When we look into this picture of the DNA strand where there is a twisted structure, we find there is a transverse, this ladder like structure. See, this is a picture of a ladder where this ladder is oriented with 5' to 3', 3' to 5'. These are the 2 DNA strands. And there are these transverse bars. These transverse bars are the pairing in between purine and pyrimidine. G to C, T to A. The nitrogen bases are present on complementary strands. They are held together by a weak hydrogen bond. Means there is a hydrogen bond. Where double bond is there in between A to T, triple bond is there into J and C. On this strand, there is a pairing of purine and pyrimidine, and it has a fixed ratio. Means there is a fixed purine and pyrimidine ratio. This is observation of Watson and Crick. What is this? According to Chargaff law, total number of purines are always equal to total number of pyrimidines. And so, ratio of purine to pyrimidine on DNA strand is always 1:1. And so therefore, A plus G, that is adenine plus guanine, is always equal to T plus C. Means purine plus pyrimidine is equal to always 1. Also we can explain A plus G divided by T plus C is equal to 1. Next property of the DNA. Next property. There is a polarity of strand. The DNA strands have the polarity. Each strand has polarity. One strand is considered as 5N dash or C5, while other is 3 dash and C3. At 5 dash end, there are pre-phosphate group. At 3 dash end, there is a pre-hydroxyl group. In DNA strand, there is phosphodiester linkage. What is this phosphodiester linkage? On each strand of the DNA, there are nucleotides. These nucleotides are present on that strand. They are always joined with each other by means of phosphodiester linkage. In this way, one nucleotide link with another nucleotide, then another to the next. In this way, there is synthesis of chain of nucleotide, and in this way, the strand get forms. According to Watson and Crick. Next, characteristics of the DNA is the strands are always anti-parallel. DNA is made up of 2 strands. These 2 strands are not only upward as well as downward direction, but they are anti-parallel. Means one strand when go to the upward to downward, another strand come from downward to upward. So they are called anti-parallel. They run opposite to each other. Then the strands are complementary in nature. Complementary means opposite. The nitrogen base sequence present on one strand is completely opposite to the nitrogen sequence present on another strand. Another property is recorded by Watson and Crick, and that property is DNA structure have major and minor grooves. Means when we look to the DNA structure, at that time there are some grooves. That groove is a deep groove as well as shallow groove. See, when we look into this DNA structure, here we find there is a minor or shallow groove, but here angle of this groove is a deep, and so it is called a major groove. Now come to the characteristics. Last characteristics according to Watson and Crick, the dimension of the DNA strand. Dimension. DNA strand, when we measure at that time, here Watson and Crick have given their observation that the diameter of the DNA molecule is 2 nanometer. See in the DNA structure. When we look into these two strands, some readings are there. So when we look into this diameter, in distance between the two strands, it is of 2 nanometer. Then, the turn of one loop. The DNA strand consists of number of loops. One loop, the second loop and third loop. In this way, there are number of loops on the DNA strand. According to Watson and Crick, each loop consists of 10 base pairs. Means these base pairs, we know, these bases are adenosine, thymine, guanine and cytosine. What are the nitrogen bases present on the DNA strand? These nitrogen bases are counted by Watson and Crick, and according to them, each loop of the DNA strand consists of 10 base pairs. So here number of these base pairs are 10. Their pairing of the bases is fixed. Means if the base belongs to purine, it pairs with pyrimidine. And we know there are two purine bases, adenine and guanine. There are two pyrimidine bases. They are guanine and cytosine. So these loop contains the 10 base pairs. These base pairs are purines and pyrimidines, and here therefore A pairs with T, G pairs with C. Then next, the distance between two successive nucleotide. Distance between two successive nucleotide is 0.34 nanometer. Means the DNA have different loops. Distance between the two loop is 0.34 nanometer. And entire turn of one loop is 3.4 nanometer. Entire turn, 3.4 nanometer. Then, according to Watson and Crick, the angle between successive base pair is 36 degree. See here, successive loop. Angle between the two loop is 36 degree. After understanding the different characteristics or the properties explained by Watson and Crick for explaining the DNA structure, after 20 years, the people were studying the DNA model as it is Watson and Crick have explained. But after the 20 years, in subsequent period of the research in the molecular biology, number of scientists have recorded their observations, and they says that DNA changes its structure. So therefore there are different shapes of the DNA, different orientations of the DNA, and so therefore After 20 years discovery of the DNA of Watson and Crick, the scientists have said that there are different forms of DNA also. Means DNA shows the variation in their structure, so it is called polymorphic. Thus, DNA can be of different types. According to them, the forms of DNA or types of DNA, they are B, A and Z. B type of DNA, A type of DNA or Z type of DNA. We have to understand the different forms of the DNA, that these scientists after the Watson and Crick, what they have studied and how they have commented. They have studied the DNA in different environment. Means percent relative humidity, the factor they have considered from the cell, or they have considered the different ionic strength, concentration of the solutions in the cell. DNA where it is present, in that what type of percent relative humidity and what type of ionic strength, means it is a lower concentration of the ions or it is higher concentration of ion, or where there is a low percent relative humidity or high percent relative humidity. In that environment, DNA is studied by different scientists and they have said there are B, A, Z type of DNA. Also they have different types as like A, B, C, D, E type of DNA, then Z type DNA. But commonly, talking on the types of DNA or forms of DNA, they are mostly explained as B type of DNA, A type of DNA and Z type of DNA. Now come to the first part or first type of DNA, B type of DNA or B form of DNA. B form of DNA, it is a right-handed. See here, most important, B type of DNA, it is a right-handed, long and thin. Helix diameter is 23.7 angstrom. Rise of base pair 3.4, base pair per helical 10, helix pitch 34, tilt of base 1 degree. Means what are the properties explained by the Watson and Crick? That orientation of DNA, diameter of DNA, base per helical, then angle, tilt of base. These properties differentiate B DNA from A from Z. See here, this B form DNA is right-handed DNA. It is discovered by Watson and Crick, and most of the living organism in their cell we find right-handed B form of the DNA. Then, how it is different from A type of DNA? Look at the A. What is the difference? Again, it is a right-handed DNA, but it is short and broad as compared to B form of the DNA. It is short and broad, but helix diameter 25.5 angstrom, rise per base pair 2.3 angstrom, base pair per helical 11, helix pitch 25 and tilt of base 20. See, this is difference between the characteristic of B form and A form. Now see, this form is different. This form is different, where B form was long and thin, another is short and broad, and Z form is longer and thinner. And one important thing, it is a left-handed. See here, first two types, B and A, they are right-handed, but Z type, left-handed. So they are zigzag in fashion. Their orientation is very, very zigzag. The Z form of DNA is observed in Drosophila chromosome. This is also one of the addition in the Z type. It is observed in Drosophila chromosomes. See here, then helix diameter 18.4 angstrom, rise per base pair 3.8 angstrom, base pair per helical 12, helix pitch is 47 angstrom and tilt of the base 9 degree. Means these properties when we compare to the B and A, these are completely different. So in this way, these are the different forms of the DNA. When collectively we study together these three different forms, and when we are starting to compare or we are looking into the difference, we find these different characteristics of B type of DNA, A type of DNA and Z type of DNA. Come to this first. Characteristics, orientation, nature, helix diameter, number of base pair, the base pair per helical, angle of the bases on the DNA strand. In the B form, B and A, first orientation, both are right-handed, Z is left-handed. Nature, first is long and thin, A is short and broad and Z is long and thinner, longer and thinner. Helix diameter, these both have here 23, 25 means near about, but Z is different. Then base pair, number of base pair 10, 11, 12. Helix pitch 34, 25, 47. A drastic change is there in the Z, 47 angstrom. And finally, the degree of orientation, 1 degree, 20 degree and 9 degree. In this way, there is a complete difference in between each type of DNA, B, A and Z. Thank you. स्टॉप करा जरा अंदाज घ्या स्टॉप केले ना नाही तुम्ही करा